They say aging is inevitable, that the body declines, the energy fades, the skin loses its structure, and the mind grows slower, all because of time. That is what most people believe. But time alone is not the enemy. Inside nearly every one of your 37 trillion cells sits a tiny structure, ancient, evolutionary, and irreplaceable. It does not simply exist. It works continuously, silently. It converts nutrients and oxygen into the fuel that powers every heartbeat, every thought, every repair your body makes. That structure is called the mitochondrion. And here is what science is beginning to understand. The symptoms we associate with aging, the fatigue, the foggy thinking, the slower metabolism, the cellular breakdown, are not the result of time passing. They are the result of mitochondrial dysfunction accumulating, undetected. And over years, the damage is silent, the progression is slow, but the biology is precise. Let's begin. The body's smallest power plant. Somewhere inside every living cell in your body, your heart muscle cells, your liver cells, your neurons, your skin cells, there are structures that have been producing energy since the earliest chapters of multicellular life. Mitochondria. They are not simply components. They are the reason complex biological life can function at all. A single mitochondrion is roughly 1 to 2 micrometers in length, smaller than a red blood cell, yet it performs one of the most energy-intensive biochemical processes in nature. Through a sequence of reactions known as the electron transport chain, mitochondria convert glucose and oxygen into adenosine triphosphate, or ATP. One ATP is the actual energy currency of the cell. It powers muscle contraction, nerve impulse transmission, protein synthesis, and cellular repair. Without ATP, none of these processes occur. Without mitochondria, no ATP is produced. A single cardiac muscle cell can contain as many as 5,000 mitochondria, an extraordinary density that reflects just how energy demanding the heart is. The brain, which accounts for roughly 2% of total body weight, consumes approximately 20% of the body's entire ATP output. Every thought, every synaptic transmission, every act of memory retrieval depends on continuous mitochondrial function. This is why the condition of the mitochondria is not a peripheral concern. It is central to everything. When mitochondrial function is high, the body operates with precision. When it begins to decline, and the decline is felt in every system, not all at once, but slowly, progressively, quietly. And the process of that decline has a biology, a mechanism, a traceable cause. The first signs the body sends long before a diagnosis is made, long before any imaging or blood work reveals structural change, the body begins to communicate. The messages are quiet at first, easy to dismiss, a fatigue that does not resolve with rest, not the tiredness that follows a long day, but a deeper, more persistent depletion, as if the body's internal battery holds less charge than it once did. The same 8 hours of sleep that previously felt restorative now feels insufficient, a cognitive slowness. Words retrieved less quickly, concentration maintained less easily. The brain, that can be the body's most ATP-hungry organ, is one of the first to register reduced mitochondrial output. Neurons require enormous continuous energy to maintain membrane potential, transmit signals, and clear metabolic waste. When the supply diminishes even marginally, processing speed is affected. A metabolic shift. The body's ability to maintain a stable weight without significant dietary changes begins to alter. Mitochondria are central to fat oxidation, the process by which fatty acids are metabolized into usable energy. When mitochondrial efficiency falls, fat oxidation slows. The body stores more of what it once burned. Skin changes occur in parallel. Collagen and elastin synthesis are energy-dependent processes. Skin cell turnover requires ATP. As mitochondrial output decreases, where the skin's structural repair slows, visible in reduced elasticity, altered tone, slower wound healing. Each of these signals points to the same origin. Not time, not lifestyle as a vague concept, but a measurable, biological decline in the organelles responsible for energy production, occurring at the cellular level across every affected tissue. How mitochondria become damaged. Mitochondrial dysfunction does not arrive without cause. It is the product of biological processes that accumulate over time, some of which begin earlier than most people assume. At the core of the damage is a phenomenon called oxidative stress. The electron transport chain, the machinery through which mitochondria produce ATP, generates as a byproduct of that very process molecules called reactive oxygen species, or ROS. When these are highly unstable free radicals, in small quantities they serve as cellular signaling molecules. In excess, they are destructive. The body possesses antioxidant defense systems, enzymes like superoxide, dismutase, and catalase that neutralize ROS before they cause damage. But when the rate of ROS production chronically exceeds the capacity of these defenses, oxidative damage accumulates. One of the most consequential targets of that damage is mitochondrial DNA. Unlike nuclear DNA, mitochondrial DNA lacks the protective histones and robust repair mechanisms that guard the chromosomes in the cell's nucleus. It encodes 13 proteins that are essential components of the electron transport chain. When mutations accumulate in these genes, the proteins they encode malfunction. The electron transport chain loses efficiency. ATP production declines. A second mechanism involves the mitochondrial membrane itself. The inner mitochondrial membrane is the site of ATP synthesis. Oxidative damage, particularly lipid peroxidation, where free radicals attack the polyunsaturated fatty acids in the membrane, compromises its structural integrity and reduces its electrical gradient. The result is less efficient proton pumping and, consequently, less ATP generated per unit of fuel. These mechanisms reinforce each other. Damaged mitochondria produce more ROS. More ROS cause more damage. A self-amplifying cycle takes hold, one that accelerates with time if the conditions that generate oxidative stress are not reduced. The factors that fail with age mitochondria do not function in isolation, or they depend on a network of molecular cofactors, biochemical assistants that enable every step of the energy production process. And with age, several of these cofactors decline in concentration in a manner that is both measurable and biologically significant. Coenzyme Q10, CoQ10, is a fat-soluble molecule embedded in the inner mitochondrial membrane. It acts as an electron carrier within the transport chain, shuttling electrons between protein complexes and enabling the generation of the proton gradient from which ATP is synthesized. Without adequate CoQ10, the chain operates below capacity. The body synthesizes CoQ10 endogenously, but this synthesis declines with age. Research indicates that by the fifth decade of life, CoQ10 levels may be approximately 50% lower than those measured in early adulthood. This decline is not without consequence. It corresponds directly to reduced electron transport efficiency and lower ATP yields per mitochondrion. NAD+, nicotinamide adenine dinucleotide, is a coenzyme that participates in the redox reactions of cellular metabolism. It is a critical electron carrier in the process that feeds the electron transport chain. NAD+ also activates sirtuins, a family of proteins that regulate mitochondrial biogenesis and DNA repair. Its decline with age, roughly 50% lower by the sixth decade compared to peak levels, has cascading consequences for both energy production and cellular maintenance. L-carnitine facilitates the transport of long-chain fatty acids across the mitochondrial membrane, where they can be oxidized for fuel. When carnitine availability declines, fatty acid oxidation slows. Less ATP is generated from fat, and the body's metabolic flexibility diminishes. These are not abstract biochemical footnotes. Each of these cofactors represents a measurable point of failure, and each decline is part of the composite picture of mitochondrial aging. The myth of irreversible cellular aging. There is a widely held assumption that what aging takes, it takes permanently. That the cellular decline observed in the tissues of older individuals is the fixed result of time, irreversible, structural, beyond recovery. The biology of the mitochondrion complicates this assumption considerably. Mitochondria are dynamic. They undergo continuous cycles of fusion, merging with other mitochondria, and fission, dividing into smaller units. This constant reshaping allows healthy mitochondria to share components with damaged ones and helps maintain the overall health of the mitochondrial population within a cell. More significantly, the body possesses a dedicated mechanism for removing damaged mitochondria and replacing them with new ones. Mitophagy, a specialized form of autophagy, is the process by which dysfunctional mitochondria are tagged, engulfed, and dismantled by the cell's recycling machinery. The components are broken down and repurposed. The space is cleared. In parallel, mitochondrial biogenesis, the generation of new mitochondria, is triggered by specific cellular signals, including activation of a transcriptional regulator called PGC1-alpha. This protein promotes the replication of mitochondrial DNA and coordinates the expression of genes involved in building new mitochondrial components. The key insight from research is this: mitophagy and biogenesis are not age-dependent events that cease to occur. They are metabolic processes that require the right cellular conditions to be activated. Those conditions are physiological, related to energy status, oxidative environment, and specific molecular triggers. Rather than fixed by chronological age, the body retains the biological architecture for mitochondrial renewal. What varies is whether that architecture is activated or suppressed by the metabolic environment the body is placed in. What silent decline looks like. Over time, when mitochondrial dysfunction is not addressed, either the downstream effects extend far beyond fatigue and cognitive fog. Over years, the consequences become structural, visible in the function and resilience of major organ systems. The cardiovascular system carries a disproportionate burden. The heart muscle, among the most metabolically active tissues in the body, contains a dense population of mitochondria. As these mitochondria lose efficiency, cardiac output is maintained only at the cost of increased metabolic strain. The heart works harder to compensate for cells that produce less energy per cycle. Over time, this contributes to reduced cardiovascular reserve and diminished exercise capacity. The immune system is similarly dependent on ATP availability. Immune cells, lymphocytes, macrophages, neutrophils, require large quantities of energy to mount inflammatory responses, proliferate, and execute the cell-killing functions that protect against infection and abnormal cell growth. Mitochondrial dysfunction reduces immune cell efficacy in a measurable physiological way. At the molecular level, the connection between mitochondrial health and telomere integrity is significant. Telomeres, the protective caps at the ends of chromosomes that shorten with each cell division, are particularly sensitive to oxidative stress. Elevated ROS levels, a hallmark of dysfunctional mitochondria, accelerate telomere erosion. Shorter telomeres are associated with cells that lose replicative capacity, a cellular level correlate of the aging phenotype. The neurological consequences compound over time as well. Neurons do not replicate. The ATP they require must be produced continuously without interruption. Chronic mitochondrial insufficiency in neural tissue is linked to gradual declines in cognitive processing, memory consolidation, and the clearance of metabolic waste from brain tissue. The body ages not because it is programmed to fail, but because the systems that maintain its cellular infrastructure progressively lose the energy needed to function. Reading the body's own repair signals. The body does not passively deteriorate, it responds, it signals. And those signals, if understood in their biological context, reveal the conditions under which mitochondrial renewal occurs most actively. One of the most studied signals is the state of energy scarcity. When cellular glucose levels fall, when glycogen stores are depleted and the fed state transitions toward a fasted state, an enzyme called AMPK is activated. When AMPK is a master cellular energy sensor. Its activation shifts the cell from anabolic processes toward catabolic and repair functions. Mitophagy is upregulated. PGC1-alpha is induced. Mitochondrial biogenesis accelerates. Heat stress produces a parallel response. Exposure to elevated temperatures, as occurs in tissues during intense physical activity or controlled thermal exposure, increases the expression of heat shock proteins. These molecular chaperones assist in refolding damaged proteins within mitochondria and protecting mitochondrial integrity during periods of oxidative challenge. Cold exposure activates a different pathway. Brown adipose tissue, thermogenic fat, is densely packed with mitochondria. Or cold-induced activation of this tissue promotes mitochondrial biogenesis through pathways involving norepinephrine and PGC1 alpha, the same transcriptional regulator implicated in mitochondrial renewal across tissues. These are not coincidences. They are the biological logic of hormesis, the principle that controlled, manageable stressors activate the body's own repair and adaptation systems. The mitochondria are central beneficiaries of this process. The body, in essence, communicates what it needs through the activation of these pathways. Fatigue signals energy deficit. Metabolic inflexibility signals a failure to switch fuels. Reduced physical recovery signals impaired cellular turnover. These are not simply discomforts. They are biological readouts of mitochondrial state. Understanding them is not merely informational. It is physiological literacy. The mitochondrion is not a detail. It is the foundation of cellular function, the origin of the energy that powers every biological process the body performs. Its decline with age is not a mystery. The mechanisms are known: oxidative damage, cofactor depletion, impaired clearance, mtDNA mutation, and the body's capacity to respond, to clear damaged mitochondria, to generate new ones, to activate repair pathways, remains present, waiting for the cellular conditions that trigger it. The body gives signals long before systems fail. The science exists to understand what those signals mean. Health is not sudden, it is cumulative, built or eroded in the invisible machinery of the cell. This channel explores the science behind everyday health topics. And if you want more simple tips to improve your health and mind, make sure to subscribe, and I'll see you in the next video.